Good evening and I welcome una back to our channel this evening. I don't tell una before say we get three-part series where we go to discuss the bacterial cells, the structure, all the things wey dey inside for the middle and outside of the bacterial cell. Today the video wey we dey shoot, we wan discuss the bacterial cell wall. Wetin dey for the bacterial cell wall. Now the bacterial cell wall dey made up of structure wey we dey call peptidoglycan, and this peptidoglycan dey very important. Na wetin we dey call polymer molecule, na giant molecule wey be say small monomer units dey there wey join together to form this giant molecule wey we dey call peptidoglycan, and e dey important to understand wetin e be. Now this cell wall, bacteria dey get cell wall just like plants dey get cell wall too, but the cell wall of plant na cellulose dey bin use make am. The one for bacteria na peptidoglycan be the material wey dey there. Now apart from say na peptidoglycan be the material wey dey there, this cell wall, part of the function wey the cell wall dey help the bacteria with na strength. E dey very strong and e dey rigid in nature in such a way that e dey give bacteria a shape. We know say many bacteria get different types of shapes. Some bacteria dey oval, some dey like rod, some dey like chain. They get different shapes, and na basically the shape wey this cell wall bin give the bacteria na the shape wey the bacteria go get. So this one dey important. This cell wall, e dey give bacteria shape, e dey give bacteria strength, e dey protect bacteria from external environment. If cell wall no dey wey go give bacteria protection, that bacteria fit just dry up easily, or external things fit come from outside come destroy the bacteria. The cell wall dey regulate, e dey manage the things wey dey come into the cell, and e dey make sure say no be everything go just dey find im way into that cell. So the bacterial cell wall dey very important, and e dey made up of material wey we dey call peptidoglycan. Another name wey we dey call this peptidoglycan na murine. Murine na another name for peptidoglycan, and this one dey important. Another important work wey the cell wall dey do na say if the bacterial cell bin find itself inside solution wey we dey call hypotonic solution. The meaning of hypotonic solution na say the salt con ten t wey dey that solution e no dey much, so there is more of water dey more for inside that solution pass salt. Now if you get solution wey water dey more pass salt, the water go wan dey find im way enter into the bacterial cell, and if e continue to dey enter, e go come dey put pressure for the walls, that's the cell wall of the bacteria. Now if that cell wall no dey strong enough, that bacteria go just burst. So part of the things wey dey keep bacteria alive na say e get very strong cell wall. Even so, if you find yourself for inside hypotonic solution wey water plenty pass any salt or any dissolved substance, the bacteria go still dey strong enough to survive. That im cell wall go dey strong. Now as I don talk before, we know say polymer dey, monomer dey, and na monomers be the small units wey if we couple am together, e go give us polymer wey be the giant molecule. Now wetin be the small molecules wey we fit couple together to make the giant molecule wey be polymer, wey be peptidoglycan for cell wall of bacteria? The small molecules wey we dey gather together, number one na N-acetylglucosamine, na the name of that monomer. Na structure wey I go show you the structure, I go show you the chemical structure, show you the diagram as e take be. N-acetylglucosamine dey. Another one na N-acetylmuramic acid. This ones na monomer units wey dey couple together to form this peptidoglycan. Apart from N-acetylglucosamine and N-acetylmuramic acid, we get proteins, amino acids. We get amino acids such as D-alanine and L-alanine. These ones too dey as proteins wey dey together inside that structure wey we dey call peptidoglycan. Apart from that, we get D-glutamate. D-glutamate na another amino acid wey dey for that structure, and the last one na A-D-amino acid. A-D-amino acid. All these monomer units together, dem dey together, couple together, join together to form that structure wey we dey call peptidoglycan layer, or we dey call murine, and e dey very important. Now we don discuss, or we don talk before, say we know say bacteria get different types. We dey call some bacteria gram-positive bacteria, we dey call others gram-negative bacteria. And the major reason why we dey get these different names for different bacteria na say their structure, the structure of their cell wall bin dey different. For the gram-positive bacteria now, they bin get very thick cell wall. Their cell wall dey very thick, much thicker than the gram-negative bacteria. This one dey very important. We go shoot a video where we go discuss, gram reaction, gram staining, come discuss all the different things wey dey for that gram staining, discuss why the thing take be like that. But e dey important to know say the different structures wey bacterial cell wall get na wetin dey make different bacteria dey inside one of the two groups. Either they go be gram positive bacteria or dem go be gram negative bacteria. This one dey very important to understand also as a student. Now, apart from this peptidoglycan cell wall wey dey for bacteria, inside the bacteria closer to the cytoplasm, we get wetin we dey call cell membrane. So the bacterial cell get both cell wall and cell membrane. Now, this cell membrane na wetin we dey call am phospholipid bilayer, and this one na because e get something wey we dey call phosphate head. The structure na say you get phosphate head and glycerol tail. I go show you the diagram, you understand. Now, that phosphate head, we dey talk say that phosphate head dey hydrophilic. Wetin that one mean na say e get affinity, e get love for water. So anything wey be philic, I don talk am for some of my older videos, say anything wey get philic, you know say e get love or affinity. If you hear phobic, you know say na hatred or you no want. So the phosphate head dey hydrophilic, e get love for water. So e dey like to attach itself towards anywhere where water dey. Meanwhile, the tails wey be glycerol for that our phospholipid bilayer of the cell membrane, the tails wey be glycerol, they dey hydrophobic, meaning say they no want water, they dey run away from water because e just be like oil and water no dey mix. Na so dem be. So e dey important to understand say cell wall dey wey dey made of peptidoglycan layer, and then cell membrane dey, and this cell membrane na phospholipid bilayer. This one dey important for us to know for inside the bacterial cell. Thank you, because I know say una don lis ten to this video and I know say una go dey understand small small. We go shoot video on gram staining and gram reaction to explain all those concepts well well. Have a wonderful evening.